Hello guys, welcome back to our channel. We are on due Pariksha and today we will be doing Unit-4, that is Citric Acid Cycle of Carbohydrate Metabolism, that is a paper of Biochemistry honors in semester 2 of DU. We will be solving PYQs of this unit. I hope आपको Unit 1, Unit 2 and Unit 3 समझ आया होगा. उसके PYQs आपको समझ आए होंगे, आपके लिए हेल्पफुल होंगे वो. अपने दोस्तों के साथ हमारी वीडियोस शेयर करिए, लाइक करिए. चलिए हम देखते हैं इसके क्वेश्चन. फर्स्ट क्वेश्चन. Before moving on to first question, I would like to say that in this unit we will be solving PYQs related to the inhibitors of the TCA cycle and its regulation and some important reactions and how it is amphibolic in nature. Now let's start with first question, that is, give the reaction for the following oxidative decarboxylation reaction in TCA cycle, and second is reaction of TCA cycle which is also a part of ETC, that is electron transport chain. If we see the first question, oxidative decarboxylation reaction in TCA cycle, this is the reaction for the first part, that is first part. इसके अंदर क्या होता है, आइसोसिट्रेट इज कन्वर्टेड टू द 2-ऑक्सो-ग्लूटरेट. It is nothing but alpha keto glutarate. It undergo oxidative decarboxylation. It means decarboxylation occurs, removal of carbon dioxide occurs, and as well as oxidation occurs. Oxidation occurs at this point. Hydroxyl group is converted to the keto group. In this, what happens? Isocitrate dehydrogenase has Mn2 plus in its active site, which interacts with what carbonyl group of the intermediate, which is formed temporarily but does not leave the active binding site until वो किस में कन्वर्ट हो जाए, अल्फा कीटोग्लूटरेट में. तो आइसोसिट्रेट बाइंड कर जाता है एंजाइम से और ऑक्सीडाइज्ड हो जाता है बाय हाइड्राइड ट्रांसफर टू NAD प्लस, और उसको किस में कन्वर्ट कर देता है, NADPH और NADH में, और खुद किस में कन्वर्ट हो जाता है, अल्फा कीटो ग्लूटरेट में, और रिलीज कर देता है कार्बन डाइऑक्साइड. दिस COOH molecule released as carbon dioxide. Now let's look at the second part of the question. The reaction that is also a part of ETC is the reaction that is catalyzed by succinate dehydrogenase that converts succinate to fumarate. What happens in this? Electrons from succinate through FAD is passed on to what? Iron sulfur centers to ETC. This enzyme is also present in IMM, that stands for inner mitochondrial membrane. Second question, TCA cycle is a amphibolic in nature. What is meant by amphibolic? Amphibolic is a pathway that has both catabolic roles and anabolic roles, which means उसका ना catabolism भी होता है. Catabolism हमने first unit में देखा था, breaking down molecules to generate energy. Anabolism, providing precursors for biosynthesis. मतलब इसके अंदर क्या होता है, इसके अंदर बिल्ड अप करते हैं हम कॉम्प्लेक्स मॉलिक्यूल को फ्रॉम सिंपलर मॉलिक्यूल्स. तो TCA साइकिल प्रोवाइड करता है प्रिकर्सर फॉर वेरियस बायोसिंथेसिस प्रोसेस. ये आता है TCA इज नॉट ओनली फॉर द प्रोडक्शन ऑफ द एनर्जी बट वही इट ऑल्सो सप्लाइज building blocks for synthesizing important biomolecules. If we look at the catabolic function of the TCA cycle, we know that when glycolysis occurs aerobically, it converts into two molecules of the pyruvate, then pyruvate is converted to acetyl-CoA. Acetyl-CoA is oxidized by acetyl is oxidized by TCA cycle, which generates NADH and FADH2 GTP molecule and two molecules of CO2, which ये NADH and FADH2 then enters ETA cycle for the production of ATP. Carbohydrates and fatty acids that gets converted into acetyl-CoA and they are oxidized by TCA cycle. Amino acids also enter TCA cycle. Now let's look at the anabolic functions. In anabolic functions, what happens? जो इंटरमीडिएट्स होते हैं टीसीए साइकिल के, वो प्रिकर्सर के काम करते हैं वेरियस बायोसिंथेसिस प्रोसेस के लिए। दे एक्ट एज बायोसिंथेटिक प्रिकर्सर। हम स्टार्ट करते हैं पहले सिट्रेट से। देखो, सिट्रेट जो एक मॉलिक्यूल है ना, सिट्रेट इज एन इंटरमीडिएट ऑफ टीसीए साइकिल। ये एक तरीके से एक्सपोर्ट हो जाता है टू द साइटोसोल फॉर lipid biosynthesis like fatty acid synthesis and sterols. Now alpha ketoglutarate is converted to glutamate. It helps in the production of the or in the it is involved in the biosynthesis of amino acid, various acids. It gets converted into glutamate, which is involved in the synthesis of various amino acids like glutamine, proline and arginine. It is also involved in the production of the purines, that is guanine and adenine. Now succinyl CoA, one of the intermediates of TCA cycle, also acts as a biosynthetic precursor for porphyrins and heme group. Now oxaloacetate, it is also acts as a biosynthetic precursor for aspartate, which gets converted into asparagine. Now asparagine, it is also involved in the production of pyrimidines, that is thymine and cytosine, amphibolic in nature because it has catabolic function and as well as anabolic role. In anabolic role, it supplies what? Biosynthetic precursors for various biosynthesis process. Now let's look at the third question. What are anaerobic reactions and give examples. These are the reactions or metabolic reactions जो replenish करते हैं intermediates of the TCA cycle that are used for various or withdrawn for various biosynthetic purposes. Example for the synthesis of the amino acid, as we have seen in previous question, it is amphibolic in nature. It has anabolic role also, that is its intermediates are used for synthesis of fatty acids and amino acids and purines, pyrimidines, porphyrins and as well as heme groups. So they are removed for various biosynthetic purposes. Or reaction ensures that TCA cycle operates efficiently for both energy production as well as for biosynthesis plus biosynthesis. When intermediates like oxaloacetate and malate and alpha ketoglutarate are drained off from the TCA cycle, these pathways restore them and maintain balance. Now let's look at the examples. First, pyruvate carboxylase. It is converted to the oxaloacetate by reacting with ATP and bicarbonate in the presence of pyruvate carboxylase enzyme to form oxaloacetate. Second is PEP, that is phosphoenol pyruvate, that also undergo carboxylation in the presence of PEP carboxykinase and utilizes GDP molecule to form oxaloacetate and GDP. Another reaction to form oxaloacetate from phosphoenol pyruvate that involves different enzyme PEP carboxylase. Here it reacts with bicarbonate to form oxaloacetate and inorganic phosphate is released. Then we have malic enzyme. What does malic enzyme do? Malic enzyme replenish करता है malate को from the pyruvate when pyruvate react with bicarbonate and undergo reduction reaction in the present by NADPH or NADH to form malate and leads to the formation of NAD plus or NADP plus. These reactions maintain TCA cycle की activity despite metabolic drain. Now let's see another question. Name the key regulatory enzymes of TCA cycle. Show how they are regulated by product inhibition and allosteric feedback regulation. So TCA cycle is highly regulated to balance the energy production with the cell's need. It is done by regulating three key rate limiting enzymes of TCA cycle. First is citrate synthase that is involved in the formation of the citrate from acetyl-CoA and oxaloacetate. It undergoes product inhibition as well as allosteric regulation. And isocitrate dehydrogenase, second one, oxidative decarboxylation में involved होता है, forms alpha ketoglutarate from isocitrate. Then we have alpha ketoglutarate dehydrogenase जो convert कर देता है alpha ketoglutarate into succinyl CoA. Now let's see the cycle. की रेगुलेटरी साइड्स हमारी ये एंजाइम हो गए सिट्रेट सिंथेज, आइसोसिट्रेट एंड अल्फा कीटोग्लूटारेट. देखो ये सिट्रेट सिंथेज यहाँ एक्ट करेगा, सिट्रेट बनाएगा. इट इज इनहिबिटेड बाय व्हाट, इट्स प्रोडक्ट सिट्रेट एंड डाउनस्ट्रीम इंटरमीडिएट सक्सिनाइल कोए जो एक तरीके से compete करता है एसिटाइल कोए के साथ. और सिट्रेट का जब accumulation हो जाएगा ना, तो these are the indicators of the high energy state. तो वो एक तरीके से एलोस्ट्रिकली इसे इनहिबिट करते हैं सिट्रेट सिंथेज को एज वेल एज अल्फा कीटोग्लूटारेट डीहाइड्रोजनेज एंड आइसोसिट्रेट डीहाइड्रोजनेज को. अल्फा कीटोडीहाइड्रोजनेज इज इनहिबिटेड बाय प्रोडक्ट दैट इज सक्सिनाइल कोए. ये जो एंजाइम्स होते हैं ना, ये रिस्पॉन्ड करते हैं टू द एनर्जी स्टेटस ऑफ द सेल. एटीपी इज एन इंडिकेटर ऑफ अ लो एनर्जी स्टेट. ये एक्टिवेट करता है, देखो किसको, isocitrate dehydrogenase को as well as citrate synthase को. एक to generate more ATP there is an additional point of regulation that is by regulation of the pyruvate dehydrogenase complex which is not a part of TCA cycle itself but regulates the entry of pyruvate into cycle as acetyl CoA. So it is inhibited by ATP, NADH, again indicators of a high energy state, then by acetyl CoA, its product, and it is activated by ADP and pyruvate. Another question is give the mechanism and inhibition of the following TCA cycle inhibitor. First is fluoroacetate. Fluoroacetate is a poisonous agent that blocks TCA cycle and it is an inhibitor of aconitase which is an enzyme that is involved in the conversion of the citrate to isocitrate via aconitrate intermediate. Okay, what happens? Fluoroacetate is activated as fluoroacetyl CoA by acetyl CoA synthetase enzyme. This fluoroacetyl CoA then condenses with oxaloacetate just like normal acetyl CoA and form with the help of an enzyme citrate synthase and forms fluorocitrate. This fluorocitrate then binds to aconitase enzyme mimicking substrate citrate and results in suicide inhibition. What? Suicide inhibition of enzyme that is aconitase enzyme, where enzyme is inactivated after binding a fake substrate. Block कर देता है conversion of the isocitrate citrate into isocitrate. ये conversion block कर देता है जिसकी वजह से accumulation हो जाएगा citrate का और halt कर देगा, stop कर देगा TCA cycle को. Let's see malonate. Malonate inhibits what? Inhibits succinate dehydrogenase enzyme of the TCA cycle. It is a competitive inhibitor, which means it resembles succinate in its structure and it competitively inhibits succinate dehydrogenase bind to its active site. इसकी active site से इस site पे bind करता है but cannot be क्या आता है but it cannot be oxidized, so ये block कर देता है succinate from binding, inhibit करता है conversion of the succinate to fumarate देन अगेन डिस्टर्ब कर देता है TCA साइकिल, TCA साइकिल इज डिस्टर्बटेड एंड क्या करता है, रिड्यूस कर देता है प्रोडक्शन ऑफ FATH2। आज के लिए इतना ही, दैट्स इट फॉर टुडेज लेक्चर। आई होप आपको समझ आया होगा अच्छे से। और अगर आपको लगता है मेरी कहीं मिस्टेक है और मैंने कोई मिस्टेक करी है और समझाने में कहीं सही से नहीं समझा पाई, आप जरूर कमेंट करिए, फीडबैक दीजिए टेलीग्राम ऐप पे, कमेंट सेक्शन में लिखिए। आपका फीडबैक और आपकी राय से ही हम चीजों को कस्टमाइज करेंगे, कैसे आगे लेके चलना है हमें। यस, यूनिट 4 से यूजली यही सब पूछा जाता है कि रेगुलेशन, एम्फिबोलिक नेचर ऑफ द TCA साइकिल, हाउ द इंटरमीडिएट्स ऑफ द TCA साइकिल आर रिस्टोर्ड एंड रिप्लेनिश्ड एंड इम्पोर्टेंट रिएक्शन। मिलते हैं दूसरे लेक्चर्स में। थैंक यू फॉर वॉचिंग आवर लेक्चर।